number 18 from your lab book, so you will need the supplies for lab number 18. Yep. And the rest of the stuff, Sarah, is in your lab kit. So the stuff on the materials list, Steve, hold on, maybe I have the wrong lab number. Let me look in my book here. Yes, we are going to be doing lab number 18. The materials you need are corn, a bean seed, and a celery stalk. You do not need everything on the list. Okay, so that is the lab we are doing, number 18. Okay, so welcome to week 5. Hope that everyone had a good week. I did take a look at your projects. They were very good. I'm going to apologize from the start. I am getting over laryngitis, so hopefully my voice will last the hour and a half and we will be all set. What we are going to do to get started today is we are going to, there were 3 of you that turned your projects in to Moodle. If you did not turn your project in, you need to make sure that I get your projects, because remember, each day late they are, you are losing 10 points. This is a class where you are getting credit. If you do not turn your projects in or your homework assignments in, you will not get credit for the biology lab. So please be sure I am getting all of your assignments. So the first person, and these are just completely random, is Gabrielle. Okay, Gabrielle, your brochure is up on the PowerPoint screen. Would you like to go ahead and share it with us, please? I'm not, excuse me, I'm not able to hear you, Gabrielle. Can we make sure that your microphone is on by going down to the wrench at the bottom left-hand corner of the screen? Go ahead and click on it and make sure the correct microphone is chosen, and or make sure that your microphone is not muted. Right. Unfortunately, Gabrielle, I am still unable to hear you. So what I'm going to ask everyone to do is just go ahead and look over Gabrielle's mushroom here, the wrinkled Rhodontis palmatus, for us. I'm not going to try to read it because of my voice today, but I'm going to give you guys a second to go ahead and look over it. All right, very good. Gabrielle did a great job talking about her mushroom here. Okay, next we are going to go ahead and go to this one, and this one, I believe this was Sarah's, but I'm not sure. So whoever this is, if you could go ahead and share with us, that would be great. Sorry about that. Go ahead, Anthony.
All right, so this is my brochure. The title is Mushrooms Delicious but Dangerous. First paragraph talks about their growth. It says, the mushroom is a type of fungus that grows above ground on soil. They are also known for their relatively fast growth, and they do this by absorbing liquids. And then I have a picture from Wikipedia, which I forgot to link on the page. And then, depending on the type of mushroom, they could live several months, while some may only live a couple of days. So this would be their life cycle. Mushrooms can be edible or very toxic. As you know, people put mushrooms on pizza and many other foods. Other types of mushrooms, however, can cause very severe symptoms if eaten. Although there are very few, some can even cause death. So mushrooms can be very good on your pizza, but do research before you decide to eat one in the wild.
All right, great job. Thank you very much, Anthony. And last but not least, this one was multiple pages. And I'm not remembering whose this was. So whoever this is, if you could go ahead and share with us.
Hello. Do you think you could put it on the second page? That's where my. Oh, thank you. Okay. I did my brochure on a mushroom as well. What is a mushroom? We all know the mushrooms that grow in our backyard after a spring shower. The mushrooms you see are examples of the fruiting bodies of a fungus, a huge system of roots and shoots working below the ground. A mushroom is only one part in this major system. In this brochure, we will be focusing on the mushroom, its history, its composition, and its use. We may even take a look at some cousins of the mushroom. When was the mushroom first discovered? Archaeological evidence shows the usage of mushrooms between ancient cultures. There are rock paintings and even some small clues that lead to scientists believing these ancient people used mushrooms for medicine and even dye. The mushroom slowly pops up again, however, over time in history through legend and recorded text. A mushroom is a member of a large group of eukaryotic organisms called fungi. The composition of a mushroom. The mushroom in our backyard is only the fruiting body of the fungus growing beneath your feet. A mushroom, in fact, burrows down and can stretch across a wide length of your yard to obtain nutrients. Channels of roots and shoots exchange nutrients and water in order to keep the fungus alive all year round. The reason why they can become so pesky is their tendency to dominate over certain plants. This is why it can be difficult for farmers or your mother's garden and even harmful. Mushrooms can be beneficial and mushrooms can have beneficial and negative uses. Some examples include poisonous mushrooms and even parasitic moss. You should be worried that the mushrooms in your front yard are not the kind of mushrooms you buy from the grocery store to put in your chicken marsala. However, mushrooms can be helpful too. In symbiotic relationships, a large tree and a mushroom can create food for each other. This is especially useful during a drought or after a forest fire. A cool fact: some mushrooms can have psychoactive properties. This includes affecting the mind through hallucinations or dreamlike states. A certain chemical called psilocybin in the mushroom induces these hallucinogenic states. They are used for medicine today to help people with psychological disorders like ADHD. However, some are sold in the black market for medicinal purposes, which can in fact be harmful. Okay, last paragraph. One big happy family. The mushroom resides in the fungus family, a wide variety of eukaryotic microorganisms. Have you ever made a sandwich? Have you ever had blue cheese crumbled over a salad? Some of the foods you eat are in fact infested with fungus. Bread comes from yeast, a type of fungus, and blue cheese is actually created by molding. Beer is even fermented using fungus. There's no need to worry though. Scientists are actually discovering more types of fungus that are helpful instead of harmful. Although there are a few out there you should definitely stay away from, it's okay to still eat your food in peace. Just remember to check the expiration date. Websites include Wikipedia and on the mushroom and the psilocybin mushroom. Special thanks to Google Images. And if you want more information on fungus, go to the National Geographic website at http:/education.nationalgeographic.com/education/media/fungi. We hope you enjoyed this informational brochure on the mushroom.
Awesome, great job. Thank you very much. And the last one is Sarah's. And here we go. All right. All right, Sarah, go right ahead.
Okay, this is my brochure, and I did it on the death cap fungus. The death cap's habitat. Death caps are normally found across Europe. They are near oak, pine, and chestnut trees, grassy fields, or woods, usually during the summer and fall. The death cap is part of kingdom fungi, phylum Mycota. And then I wrote the class, order, family, and genus. And then here are my websites that I used. And if you could switch the page. All about the death cap. The death cap mushroom is one of the deadliest toadstools in Europe. The entire mushroom is poisonous. The cap can be white to a green or yellow color with darker stripes flaring outward. The gills underneath are white, and so is the stem. There are often rings around the stem because these rings protect the gills while the mushroom was growing. The death cap is very rounded when it is younger and flattens out the older it gets. It also may have a sweet smell. Death cap is dangerous. The death cap is a very poisonous fungus. The entire fungus is poisonous. Just a small amount of the death cap can kill an adult. Symptoms of death cap poisoning can appear in 6 to 24 hours after eating the mushroom. Symptoms include vomiting, diarrhea, and nausea.
All right, very good. Thank you very much, Sarah. You guys all actually chose to do different types of fungus, which was fantastic. It was great to read about them, and for you guys to learn a little bit about a mushroom. You did your brochures a little bit differently, included some wonderful pictures. Definitely a fantastic job. Okay, so let's think about our fungus that you guys were trying to grow on bread. Does anybody have any observations that they made off of their fungus? Now, I would not expect you to get a mushroom on your fungus. However, you may have gotten a mushroom on your bread. However, you may have gotten some other types of fungus. If you got something on yours, please go ahead and let me know by raising your hand. Did you get anything to grow on your bread? Very good. Would you mind sharing, Alicia, your strawberry mold? Let me go ahead and turn your camera on. Now remember to turn your camera on. Just go ahead and click the start button underneath where the video normally comes through. And hopefully click start. Oh, I see it coming. Perfect. All right, and let's take a look at the strawberries. Very nice. There is some great mold and fungus on those strawberries. Definitely not something you would want to eat, but thank you very much for sharing with us, Alicia. If you looked at it, it fuzzy look to it, and that is the appearance of most types of fungus that are not in a mushroom form. So thank you very much, Alicia, for sharing with us. Today, Sarah has not much mold. My bread actually got really stale as well and had nothing growing on it. Part of the problem with the bread is that it is preserved with so many preservatives and chemicals that they don't grow like they used to because they are meant to stay for extended periods of time in your bread drawer, your refrigerator, wherever you are storing them. So today we are going to talk about plants. Today is our last day where we will talk about a plant type, mold, fungus, bread, and we are going to switch from today, to animals. So after today, everything that we are going to do is going to be dissections. Today we are going to talk about plants, and we are going to do a mini dissection of some plant parts. So we're going to practice using some of the skills that we will use in our dissection labs today on the plants. But before we get started, I want to just kind of review some information about plants with you guys. Now, you guys know basic things about plants already. You know plants make their own food using photosynthesis. You know that this takes place in chloroplasts using chlorophyll. But we're going to go over a couple things about just what makes a plant a plant. So if you could go ahead and look at your PowerPoint screen. I'm going to make mine larger so it's easier to see. And we're going to look at some plant characteristics and how different plants are classified. So, real quick, plants make their own food. They have chloroplasts, which contain chlorophyll. The chlorophyll absorbs energy from the sun, and the plants use the energy to make food in the form of glucose or sugar. This entire process is called photosynthesis. Here is a close-up using an electron microscope of some chloroplasts. The green circles in it are the chlorophyll. You can see the individual plant cells. Plants also have something called a cuticle. The cuticle of a plant is sort of like an armor coating. It protects the plant. It helps to keep water inside the plant so the plant does not dry out. It's a waxy layer that coats the surface of stems, leaves, and other parts of the plant that are exposed to the air. It keeps the plants from drying out. Some plants have thicker cuticles than others. What area of the world do you think plants with thicker cuticles would be found in? Where do you think a plant with a thicker cuticle is more likely to be found? Very good, Alicia, in a desert, in areas where they are more likely to dry out. Plant cells have cell walls. Again, here's a picture of a microscope looking at some plants in the circle, and the little arrow are pointing to the cell walls. So what is a cell wall? Well, the cell wall of a plant, surrounds the plant cells, and it's rigid. It's tough. It's not easy to compress. It is outside of the cell membrane, and its job is to help support and protect the plant. Another characteristic of plants is that plants reproduce with spores and sex cells. There are two stages of a plant's life. These are called the sporophyte and the gametophyte. So what is a sporophyte? A sporophyte is a plant when it is in the spore-producing stage of life. In some plants, like ferns, the spores can grow directly into an adult plant. Now, if you have ferns that grow in your yard, if you flip the fern fronds up, you may see little dots on them. They sort of look like little growths. These little dots or growths are spores. They're found under the frond leaves, and they grow directly on the plants. A gametophyte is the stage in a plant's life where it produces both male and female sex cells. Male and female sex cells must join in order for us to have a new plant. When they join, it's called fertilization. There are more than 260,000 species of plants, and these plants need to be classified. So there's a few ways plants can be classified. One way is into groups of vascular and non-vascular plants. Non-vascular plants have no pipes to transport their water and nutrients inside the plant. They depend on diffusion and osmosis to move material from one part of the plant to another. Plants that are non-vascular are very small. Examples would be liverworts or mosses, and here's a picture of a liverwort. And vascular plants have tissues that deliver the needed materials through the cells and the rest of the plant. Vascular plants can be any size, and they are divided into gymnosperms and angiosperms. Okay, what we are going to do now is we are going to look at our lab, and we are going to start getting our lab set up for today. Okay, part of the lab needs to sit for a couple of minutes before we go back to it. So lab number 18 is on page 85 of your lab book. There are 3 goals for today's lab. The 3 goals for today's lab are to observe differences and similarities between monocot and dicot plants, to view the root, stem, leaf, and seed tissues of monocot and dicot plants, and 3, to practice scientific drawings. Now you should have read the background information in here already, and hopefully you are all set and ready to begin today's lab. What we are going to do right now is only step 1 on page 86. Okay, now remember, it's good habit when you are starting a lab to put on your safety glasses. I know it may seem silly for little labs like this, but it certainly is good practice. So, in our lab, number 1, it says, fill a 150 milliliter beaker or cup, half fill with water. Put several drops of blue or red food coloring in the water so the water is brightly colored. Cut the bottom of a stalk of celery and place the stalk into the beaker. Then it has you cut the bottom part of a monocot stem. Okay, I did not put that on your material list, so you do not need to do part 2. You only need to do part 1. I am going to do part 2. So you are just going to take your piece of celery. You are going to cut the bottom part off so you have a nice fresh cut. And you are going to put it in a beaker of water with some food coloring. So go ahead and do that. When you are done, click yes. So when your celery is set, click yes. Okay, remember you only need to do the celery part, so you are just going to put, let me turn my camera back on here. Mine has a tulip in it as well. You are only taking celery and putting it into some food coloring water. Okay, and then you are going to click yes. Then you are going to click yes, just so that I know you are complete. Okay, fantastic. While we are letting that dye get absorbed into our plants, we are going to go ahead and talk about the difference between monocot and dicot plants. So before we do that, what I'd like you guys to do is see if you know any differences between monocot and dicot plants. If you know any characteristics, go ahead and write them up on the whiteboard for me, please. Okay, you should all be able to write on the whiteboard. Okay, anything that you can think of. All right, those are some great thoughts that you guys have up here. So now we're going to go ahead and take a look at which of these are correct and which maybe could use a little modification. So let's take a look at the power-up on your screen with monocots versus dicots. Monocots versus dicots. Angiosperms, which are flowering plants, are divided into two types, monocots and dicots. As the zygote grows into an embryo, the first leaves of the young plant develop, and these are called cotyledons. If you've ever planted anything in a garden, the very first leaves that break the surface are cotyledons. Your plant, all plants, all angiosperms, have either one cotyledon or two cotyledons. If the plant is a monocot, it has one cotyledon. An example of a monocot would be corn, like the kind that you eat off of an ear, a lily. Examples of a dicot would be beans, an oak tree. Just because something is either a monocot or a dicot has nothing to do with the eventual size of the plant or complexity of the flower on the plant. Okay, and here's a picture. A monocot is on the left-hand side. It looks like a blade of grass. A dicot on the right-hand side has two cotyledons, or two starting leaves. Bean plants are a great example of a dicot. Another thing to look at between monocot and dicot plants is the veination of the plant. Now I have a tulip plant here in front of me. I'm going to turn on my camera so that you guys can see it. And here I have a tulip plant. If you look at the leaves of the tulip plant, and I'm going to pull one off, and if I hold it up, Let my camera focus on it. You can see that the veins of the leaves run straight from the tip up here by my fingers to the bottom. They are a straight line. It is parallel. All the veins are parallel to each other. A dicot leaf would be like the leaf on the left in the picture here, and it is showing a veinated leaf. Now the difference is not only in the leaves of the plant, but it's also in the roots of the plant. In a monocot, there are fibrous roots, like up in the top. A bottom picture shows a dicot, which is a taproot. A carrot would be an example of a plant with a taproot. One main root going down with smaller roots coming off of it. Now flowers. A monocot flower has groups of 3. A dicot flower has groups of 4 or 5 flower parts. Inside the stem of your monocot or your dicot, you have different types of vascular bundles. In a monocot, they are scattered throughout the stem, and this is what we are going to look at in our food coloring. And in a dicot, the vascular bundles are arranged in a circle. Monocots have herbaceous stems. Dicots can have either herbaceous stems or woody stems. So for a summary of monocot versus dicot, monocots are on the left and dicots are on the right. Go ahead and take a look. Go the hell away. And here is a table comparing the two. So the lab we are going to do today is going to compare monocots and dicots as well. So let's go ahead back to our lab and let's look at, we did step number 1. So now let's go ahead and take a look at step number 2, which is on page 87. Page 87, it is asking you to obtain a corn kernel and a peanut kernel. Now, because so many people have peanut allergies, we're not going to actually use a peanut kernel. I'm going to put a picture of a peanut kernel up on the whiteboard for you. So go ahead and complete step B of number 2. It is asking you to use your scalpel. Now, if you go into your lab kit, okay, let me get my video on here. If you go ahead into your lab kit, you will see that you have a dissection kit. In your dissection kit, if you open it up, you should see a scalpel. Okay, and it will look like this. Basically, looks like a small knife. Now your scalpel is very sharp. Please be careful. You are going to use your scalpel to cut your corn kernel. In your biology kit, you will see a little tray. If you would like to use that to cut on, that's fine, but you do not need to. If you have, I just have a cutting board from my kitchen, you can do that as well. But you are going to cut your corn kernel in half, and you are going to look in it to see if you can find what you would consider a developing embryo. And then go ahead and answer question 1 and 2. While you are doing that, I'm going to put up a picture of a peanut.